for us to it's a great pleasure for us to host you in this webinar regarding licorice in COVID-19. As you all know, we have faced this global pandemic for nearly 3 years, and now a couple of weeks ago, World Health Organization uh, announced that officially announced that the pandemic is finished. So many of the limitations we had during these years are now removed, and we are trying somehow to return to our daily routine. However, this was a global important event, and it affected the humanity and human life of all of us. So I think we still need to share our knowledge and experience from different parts of the world regarding how we try to manage this disease, how we faced, and what was the outcome. So this webinar aimed to talk about one of the popular medicinal plants used in different medical doctrines all over the world, which is licorice, and how this plant could help us manage the disease. Is there a real evidence for the plant, or it is just some claims and just some traditional medicine ideas about this plant? So in this webinar, in first part, we are honored to have Dr. Keyvan Zandi as a virologist who kindly accepted our invitation, and he's going to explain the infectivity and pathogenicity of the virus SARS-CoV-2. And the second part, Dr. Mina Madda is going to explain the virtual screening studies regarding the assessment of licorice compounds against SARS-CoV-2 virus. And in the third part, I will be explaining the real-world evidence regarding the effectiveness of licorice in SARS-CoV-2. So as the first speaker, I would like to invite Dr. Keyvan Zandi. He is a medical virologist with more than 20 years of extensive research, project management experience, and proficiency in antiviral drug discovery for different viruses, including SARS-CoV-2. He has previously worked as assistant professor in Emory University, and now he is associate director of virology in Arrowhead Pharmaceuticals. He kindly accepted our invitation to join us today in this webinar. So we are all interested to listen to Dr. Zandi's lecture about SARS-CoV-2. Dear Dr. Zandi, thank you very much. It is our pleasure to have you here today, and the platform is yours.
Hello, Dr. Bahram Sultani and everyone in the ground. First of all, I would like to thank you and all the organizers for inviting me for this great webinar. And I hope that could be useful for the participants, the information that today we're going to talk about that. So I just want to make sure that I can share my slides. So that's why that I want to start with, okay, please just confirm if you can see my slides. So can you see my slides?
Yes, everything is okay.
Okay, awesome. All right. Okay. So today, for my part, mainly I'm going to focus on viral targets for SARS-CoV-2 antiviral development and kind of cellular pathogenesis of this virus, because that would be the main key, not just for this virus, for most of viruses, if we want to develop some antivirals and some cures for them. So in this case, I'm starting with some sort of introduction, brief introduction to the virus structure and genome organization. So as we all know nowadays, I mean, not just virologists, not just biologists, even everyone in the world, everyone knows that what's the SARS-CoV-2 virus. The SARS-CoV-2 is an enveloped virus with an RNA as a genome, and this virus has some sort of spikes and extracellular and extra virion proteins around the virus, which those proteins and those spikes are very important for virus attachment to the host cell. And apart from that, like all other viruses, after the virus attached to the cells and enter to the cells, then it starts its pathogenesis through the replication inside the host cell. So one of the main element and key element in cellular pathogenesis of each virus is to get to know how the virus can start the replication of genome and transcription of genome and then translation of the genes. So if we look at the viral genome organization in SARS-CoV-2, we can see that there are different open reading frames for the SARS-CoV-2 genome. And each open reading frame, can code different genes. And each of those genes, they have their own individual role, or some of them, they have even more than one role in virus replication cycle within the host cell. So the reason that I'm talking about, because these are the concept and basic element that we need to know, then we will be able to design and produce any potential antiviral, not just for this virus, for any virus. So in this case, if we look at the viral genome and if we deeply look at each open reading frame and the genes that they could be coded by that open reading frame, there are some important genes for this virus that mainly antiviral drug discovery researchers, they focused on those genes. For example, there are different proteases that this virus has, such as PL-PRO or 3CL-PRO, that you can see that each of them could be coded by the different genes, even in a one open reading frame. And there is another enzyme, which is almost the most important enzyme through the virus replication within the host cell, which we call it RNA-dependent RNA polymerase, or RDRP. And apart from that, even you can see there are some other proteins that they are also involved inside the host cell during the virus replication. So, sorry. So if you look at these genes, first of all, you can see that all of them are under the category of non-structural proteins. So not this virus again. Most of the viruses, they have non-structural proteins and structural proteins. So, and mainly the non-structural proteins are the proteins that they are involved in replication and transcription of the virus within the host cell. So we need to understand that normally in the antiviral drug discovery, we prefer to target the non-structural proteins. Why? Because then we can target the key stages of the virus replication within the host cell before the virion could be formed within the host cell. Normally, we don't want to target the structural proteins that, they are in red and you can see them here. I'm not saying that it's not possible. I'm not saying that no one does that, but normally, what we need to do for any virus that we're going to target for drug discovery, mainly we try to find the good target within the non-structural proteins, because as I mentioned again, they are involved in virus replication cycle within the host cell. So if we want to have two categories, the genes with the high priority as the targets for antiviral discovery for SARS-CoV-2, and the genes and the targets with lower priority. So the genes with higher priority, we can see them that they are categorized and they are, prioritized in a way that I put it in this slide. You can see that some of them, are more important than the others. So you can see the rank order here from the top to the bottom. So for this virus, RDRP or RNA-dependent RNA polymerase is the most important gene for as a target for drug discovery, because that gene and the production of that gene, which is the RDRP enzyme, is the main enzyme that the virus needs to use within the host cell for the virus replication and genome replication and transcription. And the other protein, which many companies or academia, they target that also, is are actually two proteases of this virus, which one of them is 3CL and the other one is PLpro. And we can see that even we have drug nowadays for them. So the protease genes, they are very important. Later in the slides, I show that how they are important. But we need to understand that, according to the strategy of this virus for replication within the host cell, this virus make a polyprotein first, and that polyprotein should be cleaved by viral proteases to make the smaller proteins and functional proteins, which means that without these proteases, that cleavage couldn't be happen. And if that cleavage won't be happen, then the proteins are not functional, which means that the virus replication could be aborted. So the other enzyme which is important for RNA replication and transcription, again, is RNA helicase. We all know about the helicase enzymes in the biology. So this enzyme has the same role that we know for any other helicase, but this particular helicase is the only helicase that the virus needs for unbonding the RNA and the genomic strands to start and make the viral genome ready for replication and transcription. And the last but not least is about targeting the viral RNA itself. Means that apart from targeting the particular genes, we can target the viral RNA itself as well, which usually we do it through the RNAi or siRNA technology or antisense oligonucleotides to target the viral RNA, and eventually we can abort the virus replication within the host cell. So there are other targets, viral targets also in SARS-CoV-2 that they could count as the target, but with a lower priority. For example, the spike protein of the virus was from the beginning was one of the targets, but to be honest, there was no great success. Not for this virus, as I mentioned at the beginning of my presentation. Not for this virus. For most of the viruses, we don't want to just target the spike. There are reasons for that, because first of all, the spike proteins are the most. The external proteins for the viruses, they are the most vulnerable proteins for the change and facing to the mutation, which we saw it for COVID a lot, and that was the reason that so many monoclonal antibodies at the beginning, they got approved as a drug, but then after a while, FDA halted them or dropped the emergency use authorization for them because they found that the spike got some sort of changes, and then those monoclonal antibodies, they cannot work anymore. So the other target could be the ACE receptor, which we cannot call this as a viral target, but in any case is a target, is a host cell target, because ACE2 is the main receptor for SARS-CoV-2, but that one also has some sort of problem, because there is at least one very well-known alternative receptor for the virus, which is TMPRSS2, which later I'm going to talk about it. And the last gene that some people, they targeted during the pandemic was the N protein or nucleocapsid protein, which some people, they focused on that because that gene also has a role in assembly of the virus within the cell and could be a target, but as I mentioned, with a lower priority. So if in this diagram, if you look at the virus replication cycle, which I'm calling it always cellular pathogenesis of the virus, you can see that after the virus attached to the receptor, which let's talk about the ACE2 receptor, then we know that the virus needs to move to the cell. So that's why one of the points that could be targeted could be this stage, which I was talking about, ACE2 or spike. But then after the virus getting into the cells, so because the virus and the nature of this virus is that the virus goes through the receptor-mediated endocytosis. So then the virus needs to be released from this endosome. So that's why that again, another target could be aborting an inhibition of this release from the endosome for the virus. For this one, we can focus on fusion inhibitors as one of the classes for antiviral drug discovery. But let's say, apart from this, let's say if the virus will be able to be released from the endosome, then definitely after uncoating, we're going to see the mRNA and then genomic strand that is going to be transcribed and make an mRNA, and then that's why that's the other place that we really want to target that. And that's the place that you can see that some other players coming to the stage, for example, the protease of the virus or the RDRP of the virus. So, and as I mentioned, those are the main places that we want to target. So how we can target them? By inhibitors for viral protease or by inhibitors for RDRP, which one of the main classes that we have remdesivir for that is the nucleoside analog class that not just for this virus, for many other viruses, people can use it. So you can see that in this diagram, mainly where are the main targets for the virus replication within the host cell. So from now, I'm just going to focus on some, let's say, unsuccessful efforts for antiviral drug discovery for SARS-CoV-2, which at the first place they showed very promising, but at the end they were not successful. For example, so if you look at the history of chloroquine and hydroxychloroquine, I'm 100% sure all of you guys heard around the beginning of COVID about these two compounds, these two drugs, and people were thinking that, hey, we can repurpose these drugs for this virus and we can use it. And many people showed even in their papers that those drugs are very active in vitro. But why? Because as you see in this diagram, there was a very well-defined mechanism for these two compounds to inhibit the release of the viral particles from the endosome within the host cell. And so that's why that people were thinking and were starting even some places to use that. Even FDA at the beginning allows some sort of studies as a clinical trial. So many papers in a great journal such as Lancet, they came out and they said that it could be a good drug. But after a while, they understood that no, that's not the case, because the virus not just only use the ACE2 receptor, but also use the TMPRSS2 as an alternative. And then there was a time that this paper came out, and it shows that hydroxychloroquine couldn't be couldn't be a good solution for SARS-CoV-2 because of the TMPRSS2 expression on the host cells. So and that's why that you can see so many controversies in terms of papers, in terms of news, that all those news were in the media all the time, that, hey, we can use it, or no, we shouldn't use it. And, you know, at the end, even the paper that just published on Lancet at that time, they retracted because they found that, oh no, it cannot be used as a real drug for SARS-CoV-2. What I'm trying to say is, we shouldn't target, and we shouldn't just put all our efforts and put all our eggs in one basket for drug discovery for such a important virus, just to focus on you know, the initial steps of the virus replication, which is going to be the attachment to the cell and entry. So, and I'm just briefly want to talk in this slide about the main class of viral polymerases inhibitors, which they were in the you know, in the market since many years back. The nucleoside analogs, actually what they are. So if we talk about it, we all know what is nucleotide, okay? But what is the nucleoside analogs? You know, from the name, we can understand that they are mimicking the nucleotide, but they are not the real nucleotide. Actually, we can call them, they are fake nucleotide. So the way that they look like, you know, if you look at this figure at the left side of my slide, you can see the nucleoside analogs, you know, compatible to nucleotide. They have their own sugar, they have their own base, but they have modifications that they are in red here. So we can make so many modifications through the base or sugar, which eventually that could make this molecule very similar to nucleotide, but non-functional. And that's the way that, you know, so many drug candidates came to the world, not just for COVID, for many other viruses. Some of them were successful, not all of them. And, you know, the way that they act, you can see here in the right-hand side, while that RNA polymerase of the virus start, or even DNA, could be DNA polymerase for DNA viruses. When it starts, you know, replicating or transcribing the viral genome, so through that, because they need to use the nucleotide pools from the host cell, then these fake nucleotides could come over and come to the place, and then by mistake they can put it inside the any of those chains, and then you can see that then the extension couldn't move forward, and there will be an error. So, which means that the replication or transcription could be aborted. But we need to understand this class of drug, they have a lot of side effects, because even the polymerase of the host cell also could make a mistake, and by mistake they take it. But they are trying to improve these structures always. And you can see the remdesivir came to the place for SARS-CoV-2. I'm not going to talk about that because it had also its own caveats and up and downs. But in any case, that was at that time the only available drug. So here I'm trying to show that how the protease inhibitors work in general. So the protease inhibitors, they came to the world long time back as antiretroviral for HIV or anti-hepatitis C virus, and most recently for SARS-CoV-2, which we know the Paxlovid for that. So, and then if you look at the way that how they work, actually those protease inhibitors, you know, like the Caesar, they are very important because they, you know, their role is really needed for the virus replication cycle to cleave the polyproteins and make the smaller proteins. And then those smaller proteins could be functional, as I mentioned at the beginning, but we need to understand the inhibitors could inhibit this process. So this is one of the work that we have done the time that I was at Emory University. Why I put this one here, because I know that today we're going to talk about the botanical you know, compounds that they could be used as virus replication cycle inhibitors. At that time, we have done something on bicalane and bicalene, which they are, as you can see here, they are polyphenolic compound with the you know, botanical origin from one of the important medicinal traditional medicine from in China. So, and then we discovered that those two compounds, they could be very active, as you can see in this table, against the SARS-CoV-2 replication within the host cell. And even mechanistically, here we showed that where they could be, effective, which we showed the effect of the virus could be on the RNA-dependent RNA polymerase of the virus. Sorry, yeah, the compound has that effect against the RDRP, and it was very clear here in this gel electrophoresis. But I just want to tell you that just around the time that we were working on that, some other people showed a strong evidence that these two compounds also could have the effect as protease inhibitors. They also had a strong evidences. I'm not going to say that they were wrong or we were right, but what I'm trying to say is for botanical compounds, we should be very careful because they can have different effects against the different genes and against, even some cellular elements. And it is important because in a way, maybe we say, oh yeah, that's great because it could affect the 2 or 3, targets. But at the meantime, in the real world, especially in pharmaceutical industries and in the real world for development of antivirals, what we need and what is desirable is just to show that your drug or your drug candidate could affect one target, not so many targets. If you show that, then there could be a concern which could be raised that, hey, maybe it could have some off-target effects as well. So that's why that I just want to show this as an example. Just we need to be cautious and be in the safe side when we talk about the botanical compounds. So, and then by this, I'm just finishing my presentation. I'm sorry if I'm over time. So I tried my best to be very, fast. And if there is any question, I can stay in the call for a few minutes.
Thank you very much, dear Dr. Zandi. It was a really interesting presentation, especially the last part, which is my field of study on botanicals. I want to ask you a question which is personally important for me. As you as a virologist and someone who has worked on different antiviral compounds, both from the so-called Western medicine or conventional medicine, and also phytochemicals, how do you see the future of development of natural antiviral compounds based on phytochemicals? Do you think this is a correct way to follow the antiviral compounds from natural products, herbal medicine, or other natural sources, or we should put our focus on medicines like antibodies or biotechnology medicine? What is your idea in this regard?
Well, as a person who started my work in the, world of antiviral with the natural compounds, I'm kind of, leaning towards the natural compounds from the beginning. But the reality, we need to think of reality, So the reality is for botanical compounds, if you talk about the crude extract, or if you talk about the the compound from a crude extract that can show, different effects in the virus replication cycle, then, I'm a bit cautious about that because I saw those things, they never moved forward. As I mentioned, if you can have a compound from, doesn't matter, it's from botanical root or any other thing, but you can show specifically that compound is on target, the off target is low or zero, and you can just target this specific viral element or host element, because it could be against a host factor, doesn't matter, but as long as it doesn't show cytotoxicity, not just in vitro but in vivo and even in human, that's something good. The other thing, some people try to say that, hey, this plant extract is really great and we show this effect, and we can use it as antiviral. But the problem is you need to understand that what element was the effective factor and what's the percentage of that element within the whole crude extract. So if the element that you defined and you identified in that crude extract is just 1 or 10% of that, the whole crude extract, then you need to realize how much of that extract you should give to the patient. First of all, to be bioavailable, because the other problem is about the bioavailability of those compounds. These are the issues that you need to think of it, especially the bioavailability, because if the drug itself, drug candidate itself is not very bioavailable, how you can make it bioavailable? One of the problems that we had with bicaline and bicalane was the same thing, because I was working in those compounds since many years back for different viruses. So they are great, but they are not that very good compounds in terms of bioavailability. So, I mean, yeah, if your compound is something that is bioavailable and you know, it is right on target, I'm saying yes, totally. Yeah, you can move forward. But the problem, if you in reality look at most of those things, you can see that they are not very bioavailable unless you find something unique.
Thank you very much. That was a great answer. And actually, it's a completely complex process discovering new drugs and active ingredients.
Yeah.
So I see one hand rise, Dr. Ahmed. I think he's going to ask a question. So
yes, go ahead.
Yeah, we're ready, Dr. Ahmed. Okay, it seems that it was wrongly, his microphone was wrongly turned on. Okay, right. Any questions from other participants, please? From this part, which was the first part of the webinar. So thank you very much, Dr. Zandi. It was really great, and this presentation provided a brief but complete background regarding what SARS-CoV-2 does to our body and how we should try to react to it. And that would be a good actually base for the next speech by Dr. Mata. So I say bye for now to you, and we're gonna go for the next part of the webinar.
Thank you so much again, and have a great day. Bye.
Thank you very much. Bye. Let me see just if Dr. Mata is now available for presentation, because it was a small technical error at the beginning of the webinar. I hope the problem is solved right now.
Hello, do you have my voice?
Hello, Dr. Mata. Yes. So hopefully we are finally having you here.
Yes, because the software of the Zoom in my laptop was not updated, and the internet doesn't allow me to update at this time. And I compelled to present my slide from my cell phone, and I think it's sometimes problem. I start to share my
So meanwhile, let me just provide a brief introduction of your previous experience. Dr. Meena Mehta was a graduated with bachelor's degree in applied chemistry, and then she continued her education to master's degree and PhD of physical chemistry in Khaja Nasir Tusi University of Technology in Tehran and Iran. Her field of expertise include molecular dynamic simulation and molecular docking. She has recently worked some joint researches with medical universities on the antiviral activity of natural products. And today she is going to tell us about the simulation of licorice compounds against SARS-CoV-2 virus. So we are ready to listen to your interesting lecture, Dr. Magda, and the platform is yours.
Could you please show my slide by administrator, because I cannot do it on my phone.
Ms. Sadeghi, I colleague, yeah, thank you very much.
Okay, thank you. Thank you very much. First, thank you so much for inviting me and give me this time to present my or the research here. This presentation is about using computational methods and computational methods for drug development and drug design, and we want to describe one of our works on this method that we wanted to propose, and potential antiviral agent from the licorice compounds against the SARS-CoV-2. Today's strategy of the drug design can be classified into three groups, and the first one using and testing the broad spectrum the drugs against new targets. The second one utilizing the different computational methods and methods to screen the different databases such as natural products or drug databases to find an effective compound. And the third one is using the directly genomic information to develop a drug. And we want to use the computation method as it before for proposing the antiviral against the SARS-CoV-2 from a natural product of the licorice. Today's computer-aided drug design allow compounds to be screened computationally before testing experimentally. Therefore, it can save time and cost. Could you next slide, please show me the next slide. Thank you. SARS-CoV-2 is an RNA virus consists of structural and non-structural proteins. And for this study, we selected 6 targets play critical role in the viral replications and infections. They are main proteins, papillary proteins or pro-proteins, hexameric endoribonucleases, RNA-dependent RNA polymerase, and receptor binding domain from the spike proteins. And also we selected a target from the human cells, the angiotensin converting enzyme 2 or ACE2. So for this study, next slide. Thank you. Today there is a different and a lot of research on the screening different databases to find the pharmaceutical property of the different molecules. Licorice is one of the natural and medicinal plants that extracted from rhizome, dried rhizome of this plant, and total extract or isolated phytochemical of these compounds show a different antiviral property, anti-inflammatory and antioxidants. And these compounds extensively used in traditional Persian and Chinese medicines. And for this study, we gathered different bioactive compounds and metabolites of these plants, and almost 56 licorice phytochemical were selected, and you can see the names of these phytochemical in the table for starting the starting or computation and or methods. Next slide. Thank you. We use structure-based visual screening to propose antiviral agents against the 6 selected targets from the natural product of the liquids. You can see the flow chart of our applied method in this slide, and I will explain each stage by detail in the following after my presentation. Next slide. The first steps in the structure-based visual screening was molecular dockings. Dockings can predict different binding mood of a ligand and the affinity of a ligand in the complex receptors. The scoring functions of molecular docking, which is a mathematical function, can determine the binding force of ligands and its energy and or affinity of special ligands in the receptors. And in this study, we want to use the molecular docking for finding the potential compounds from the database of the liquid against each target of the of our selected target. Next slide. Thank you. When a ligand docks to a receptors, and in this case, a ligand docks to a protein, ligands can possess different arrangements and can move along the proteins, and each of these positions and its orientation and arrangement of the ligands named docking post. And in this slide, I show for a ligand different position or different docking posts by different colors, and you can see a ligand can form interaction with the binding site of proteins or with the amino acid of the proteins, and for each pose, the different interactions and the different amino acids maybe take part, and each docking pose that have a lower docking energy is the best for the this ligand in the binding site of the proteins. And the main aim or the main goal for docking is finding the best binding pose of the ligands in the binding site of the proteins. Next slide. Thank you. The first step for docking is finding the 3D structure of the protein or initial conformations of the proteins. We use the protein data bank database PDB for finding the initial and 3D structure of the protein with the following PDB codes that's shown in the table. And each, you can see the secondary structure of each 6 selected targets. Moreover, we need to know the binding site of the proteins or the key amino acid or residue that participates in forming the binding site. And we obtain the information of binding site from the literature or physiographic data. In this slide, I show the binding site of the proteins with the green colors, and if the administrator tap in the slide, you can see the green circles around the binding site of the proteins. Please, next to the slide. Thank you. Next. Okay. When a protein extracted experimentally, there are a lot of ligands, ions, and water molecules in the structure of the proteins. We need to remove them before starting the computations. And also some proteins have a missing residue or amino acid in their structures. We need to add a model of these regions, these missing regions before computation. Moreover, we should add the hydrogen atom to the amino acid of the proteins and the proteination state of the amino acids, especially the binding site, should be checked and assigned at a special pH that's reported, or if you cannot find any information about the proteination state, you can set the pH 7. And moreover, we need to know about the binding site of the protein, because some protein, the binding site placed between two chains or two monomers. And in this case, we need to keep all the chain and monomers for computations and for simulations. But if the binding site of the proteins placed in one monomer or in the pocket of the one chain, we don't need to keep all the chains and monomer. For example, hexameric endurance NSP15 that's shown in this slide is conformed 6 monomers, and for this study, since the binding site placed on each monomer, we just only use the one chain for a study. But in cases that the binding site placed between two chains, we need to model and keep the change for a study. Next slide. The structures of the licorice phytochemicals were retrieved from the PubChem database with the hydrogen atoms and the isomeric form and the tautomer states of the licorice compounds were checked and controlled by literature and chemistry and different packages. The structure of the phytochemical were minimized and optimized, and all of the structure needs to be in the 3D or three-dimensional format before start the computations. And we check and all these parameters about our ligands, and then the ligands are ready for computation. Next slide. We use Autodoc and Autodoc tools for preparing the input file and dockings. The following setup was used for docking simulations, and we defined the grid box of the molecular dockings around the binding site of each protein. And you can see the dimension of the grid box and also the center of the box in the table for each of receptors. And the dimensions or the volume of the grid box was large enough to allow a ligand can move freely in the box of the docking. And the reported value for each receptor was used for start the docking simulations. Next slide. One way to judge about the docking performance and accuracy of the molecular docking is by redocking a control molecule in the binding pocket of the protein. Originally, a control molecule is any kind of the ligand or compounds that present in the PDB structure initially, and we extract them and redox them again in the binding pocket of the protein. And RMSD or root mean square deviations provide a criteria for evaluating the accuracy of the docking. And according to the RMSD between the crystal structures or crystal pose and the predicted pose or the pose that's obtained from the dockings after redocking, we can confirm that whether or not a close match duct pose was predicted by docking. And the low RMSD of the redocking indicates the good performance of docking simulations. And in this slide, you can see the control molecules for 5 of receptors or target receptors, and the name of and also the RMS obtained value of the RMSD for each control molecule. According to the value of obtained RMSD for each control molecule, we ensure that our performance of the docking is good enough. Moreover, the obtained docking energy from the redocking control molecules define cutoff energy for choosing and selecting ligands from a large database. For example, we can choose those licorice compounds with a lower docking energy than control molecules as probable inhibitors or probable compounds with the desirable actions or desirable functions. And the obtained value of the docking energy for each control molecule was also reported in this table. So please, next slide. Next slide. Do you hear me, or do you lose my voice?
Yes, I have your voice. Oh, it's okay.
Okay, because I didn't see the next slide. So thank you. The first steps in virtual screening, therefore, was molecular docking. And in this step, we dock all 56 licorice compounds on each receptor and each target. And then we select those licorice compounds that have a lower docking energy or higher negative docking energy than respective control molecule on each target. And in this step, according to the value of the docking energy for 56 licorice compound, almost 15% of licorice compounds were screened and select at this step. Please, next slide. Next. Thank you. A major issue in the structure-based virtual screening is considering the flexibility of the protein during the docking. We know that during usual molecular docking, ligand is flexible and it can change different orientation and positions, but the protein or receptor is rigid and the amino acid of the protein cannot move during the docking simulation. Therefore, we need to perform ensemble docking to ensure about that the flexibility of the proteins doesn't affect on the result of the virtual screening. Several protein crystal structures that isolated or co-crystallized with various ligands construct an ensemble. This ensemble of these protein, these PDB structures can indicate a flexibility for the protein. And if we dock all 56 liquid compounds on these ensembles and compare the results after the screening, we can conclude that if the flexibility of the proteins can change the results or not. For this reason, we select two proteins, MPERO and PLPERO, for ensemble dockings and checking the flexibility. We can in this table you can see the PDB ID for selected for the PDB ID that's selected for the MPERO and PLPERO. For MPERO, we select three PDB structures. One of them is apo enzymes and two of the holo. For PLPERO, we just select two enzymes, two PDB structures, and apo and a holo. All 56 licorice compounds dock on each of these PDB, and finally, based on the statistical analysis and statistical tests, and we concluded that the flexibility of the proteins doesn't change the result of the screening test. And according to the similar trends of the docking energy, you can see that no significant changes was observed in the docking energy by considering the flexibility. Next slide, please. Thank you. In this slide, three PDB structure of the MPRO that used for ensemble docking was shown by different colors. You can see the co-crystallized ligand for each PDB change the structure of the proteins and also the positions of the amino acid in the binding site. And if we dock all 56 liquid compounds on these different binding sites, we can consider the flexibility because the co-crystallized ligands form different interactions with the amino acids of the proteins, and it change the positions and also the orientations of the binding site. And for this reason, we should ensure about the results of the effect of the flexibility on our results. We perform ensemble docking. Next slide. Next slide. Thank you. In the second step of virtual screening, we perform AD-MET-SAR and we screen out all 56 liquid compounds again based on the physiochemical property and drug likeness. At these steps, all physiochemicals were classified into three groups of the good, acceptable, and need to manipulations. And at the second step of the virtual screening, we just select and choose those liquid compounds that have a good and acceptable property. After these steps, some of the liquid compounds were removed from the virtual screening, and moreover, after the final, after the end of the second step of the virtual screening, we choose the common licorice compounds that have satisfied the thresholds of the two applied filters, molecular docking and AD-MET-SAR. There are only 13 licorice compounds that can pass the threshold of the docking filters and AD-MET-SAR filters. And in these 13 licorice compounds were further screened in the next steps. Please, next slide. Thank you. In the third step of the virtual screening, we consider the interaction profiles between the amino acid of the binding site or amino acid of the protein with the 30 licorice compounds that selected in the at the final of the second step of the virtual screening. We observe that many hydrogen bonds form between the amino acids and licorice compounds. The amino acids shown with the blue colors and the licorice compounds was shown by brown, light brown, and also different kind of the hydrophobic interactions also form between the protein and licorice, those 30 licorice compounds. And we found that most of these interactions can stabilize the binding of the licorice in the pocket of the protein. And at this step, we select those liquid compounds that can form more interactions with amino acids and the liquid compounds that form interactions with the key residue of the binding site. And based on these two applied filters at this step, only 6 liquid compounds were selected for the third step of the virtual screening. Next slide. Thank you. You can see the chemical structure, name, and abbreviation name of the top 6 liquid compounds. After the third step of the virtual screenings, 4 of them are from flavonoids and 3 of them from terpenoid group. And we use these 6 liquid phytochemicals for MD stimulations or molecular docking stimulations in the 4th step of the virtual screenings. Please, next slide. Thank you. Therefore, we conducted the all-atom molecular dynamic simulations on 6 phytochemicals, six top phytochemicals selected from the virtual screening in complex with each receptor or each target. And we use the AMBER 20 program package for simulations, and following setup was used. After the simulation, we can obtain many structural dynamical properties of the systems from the MD simulations that can help us to understand the different interactions take place between proteins and ligands during the dynamic and also in presence of the solvent. Next slide, please. Thank you. The first parameters after the MD simulations need to consider is the gross minute square deviations or RMSD, because we need to know about the equilibrations of the system, and also we need to know about the stability of the protein. Because whenever a small molecule or a ligand binds to the protein, it can make remarkable changes in the protein structures and protein conformation. And RMSD is a very good parameter that give us overall information about the stability of the protein. And we computed the RMSD for each proteins at the final of the MD simulation, and we compute the RMSD for the backbone atom of the proteins and compare the RMSD with the initial structures. And we understand that the presence of the phytochemicals in emperor has lowest effect on the structure, because according to the RMSD, the trend of the diagram is uniform, and the fluctuations for emperor is low, while on the other side for PL probe we see very high fluctuations and ununiform trends, and also the the effects of the phytochemical or solvents during the time of the simulation can cause these changes in the structure of the protein. And in this aspect, we can conclude that the simulations of the proteins in presence of the solvents during the time how change the structure of proteins. Please next slide. Thank you. The other parameters that need to consider and provide very good information about the binding small molecules into the protein is the end-to-end distance. We measure the distance between the center of mass of proteins and ligands during the simulation time to find that how the ligand molecules change its orientations or positions during the time and in presence of the solvents, because the solvents can change the position of the protein, and also in during a line time, because the different inter and intramolecular forces, the positions of the ligand molecules can vary. And these parameters could show us how ligand molecules act during this process. According to the obtained diagrams for the end-to-end distance, we can see some ligand molecules showed ununiform and with a high fluctuations trend in the diagram, and these changes is not so much, almost 3 or 2 angstrom, but for ensure about that the ligand molecules doesn't leave the binding site of the protein or doesn't leave the pocket of the protein, we need to check the a snapshot of the MD simulations or the structure of the proteins at a different time. Next slide, please. Thank you. We extracted an initial and final snapshot from MD simulations, and to consider the positions of the ligands during the simulations. And in this step, you can see the licorice phytochemical doesn't leave the binding site of the protein, but they move slightly during the pocket of the proteins or during binding pockets of the protein. And in some cases, they slightly change its orientation compared to the binding site, but they never leave the binding pocket of the protein. And by checking the snapshot of the simulations at a different time, we just show here the initial and final, and which we can check the different time of the simulations and show and find how the licorice compounds or any ligands move or act during the simulations. We can ensure that the ligand molecules don't leave the binding pockets. Therefore, at this step, the ligands can change the functions or can inhibit the pocket of the proteins, and it is what we need for designing an inhibitor for enzymes. Next slide, please. Thank you. And the other parameters that need to check after the MD stimulation is the interactions and different kinds of the interactions that form between ligands and proteins. We can measure and we can count the number of the hydrogen bonds that form between amino acid and proteins, and between amino acid of the proteins and ligands during time. And also with the 2D interactions profile, we can show different interactions that form between ligands and proteins. We observed that most of the selected Licorice compounds form many hydrogen bonds with the amino acid of the proteins. They are shown with the green colors, and also hydrophobic interactions between the hydrophobic parts of the ligands and amino acids of the proteins also observed. They are shown by pink colors. And for some ligands, we observe unfavorable interactions that are shown by red colors. And this interaction displays that the binding of the ligands in the protein active site or pocket of the protein is not suitable, and the ligands maybe tend to leave this position, and it is not a strong bindings, and we need to check these interactions, it is unfavorable interactions for all ligands, and at this step, some of the ligands after the end stimulation were removed from the 6 selected compounds, and for the as a final analysis after the MD simulations as a final parameter after virtual screenings, we calculate the binding free energy to exactly compute the binding free energy of the ligand in macromolecules of the proteins. Next slide, please. Thank you. The obtained binding free energy from the MMGBSA method is more accurate than docking energy. It also can consider the solvent effects and the dynamics of the environment during the calculations. And MMGBSA also can highlight the different contributions of the binding free energy, for example, van der Waals electrostatics, and the value of the binding free energy and its components are reported in this table. The obtained value for free energy displays that main protease has a higher negative free energy among the 6 selected targets with the licorice phytochemicals. It means these protease have a higher tendency for bindings of the ligand. On the other side, receptor binding domain has a higher positive of the binding free energy. Also, all the binding free energy are negative value, but the value for the receptor binding is higher in a positive value. And it means that receptor binding domain is not a good proteins or good targets for licorice compounds. And also we found that among the 6 selected licorice compounds, one of them have a higher negative binding for energy than other 6 licorice and other 5 licorice compounds, GCA or glyceric acid. form the high interactions energy or high binding to energy with most of the targets. And at this step, based on the obtained docking, based on the obtained free energy, we again screen these 6 selected phytochemicals, and one of them, which is the GCA or glyceric acid, proposed as the best compound for inhibiting of most of the targets. Next slide. Thank you. As a conclusion of this paper, we performed virtual screenings as a computational method to find potential antiviral against the SARS-CoV-2. According to the different analysis and different computational methodology that we applied during this study, we found that main protease is enzyme that can have a very binding affinity for the licorice compounds according to the obtained energy from the dockings, from the MMGDSA, the interaction profiles from the MD simulations, number of the hydrogen bonds. This target is has a high tendency for binding the licorice. And the GCA or glyceric acid is one of the best and effective compounds for inhibiting or binding to the selected target was chosen. And we can finally propose this phytochemical as an probable compound with the antiviral activity for further in vitro and in vivo studies. Thanks for your time and attention. My presentation is finished. Thank you.
So many thanks, dear Dr. Mata. Your presentation was actually the core of this webinar, which explained how these different compounds from licorice can interact with SARS-CoV-2 proteins, and how it is probably able to actually prevent the infectivity of the virus. And as someone who was involved in the project personally, I know that it was a really heavy work, and all these diagrams, all these figures and images you show took a lot of time to get produced. So it was a really valuable work, and actually you could perfectly represent it, and I'm sure that all the dear audience were really actually satisfied, and they enjoyed your presentation. So we're gonna have a couple of minutes if anyone has a presentation, anyone has a question about Dr. Mata's presentation. So Dr. Mata, can we have you up to the end of the presentation? I mean, up to the end of the webinar, which would be, I think, in less than half an hour.
Yes.
So that would be great. So our audience can compare the results of virtual screening with the experimental data that now I am going to represent, and that might be something actually interesting to see how those virtual screenings are reflected in real world evidence.
Yes, thank you.
Thank you very much again. And at this part, I will be happy to deliver my own presentation as the third and last part of this webinar. So let me share my screen and so that you can see my slides. Okay. Would someone please tell me if the slides are visible so that I can start my presentation?
Yes, doctor, it is visible. Could you make it in full screen mode?
Yes, thank you very much. So, okay, my name is Ruda Sultani. I'm assistant professor of traditional pharmacy and also vice dean for international affairs at the School of Persian Medicine, Tehran University of Medical Sciences. I'm really happy to be part of this webinar, and I hope this last part of the webinar can complete the other parts, the previous two parts, which together would finally answer our question about whether licorice would be something helpful for COVID-19. So up to now, we understood that SARS-CoV-2 how SARS-CoV-2 can cause infection to the human body and our cells, and that according to the results of virtual screening, the licorice compounds are probably able to inhibit some of these protein interactions and can be effective about management of the disease. But let's take another look at this plant. About taxonomy and botany of the plant, the scientific name, as Dr. Maddah mentioned, is Glycyrrhiza glabra. This is the official species of licorice, which we use in Iran, but in some other countries, other species like Glycyrrhiza urulensis are also used. Second species is mostly called Chinese licorice. The English name, as you see, is actually that licorice, and in Chinese it is called gankao, and in Indian it is called mulaiti. I'm sorry if I'm pronouncing the Indian name wrongly, because I know we have a lot of Indian audience. So in Persian, we call this plant as Shirin Bayan, and as you see, the medicinal part of the plant are the roots with a light yellow inner part, having a sweet taste when chewed, and it also has a bitter aftertaste. So if we take a look at geographical distribution of the plant, it currently can be considered as a cosmopolitan plant because it is available in all parts of the world, but it originally is grown in Iran, India, China, different parts of Asia. So it is not far-fetched that these countries use this plant a lot in their traditional medical doctrines like traditional Chinese medicine, Persian medicine, Indian medicine, so on. As previously mentioned, the major phytochemicals of licorice are categorized into two main classes, including flavonoids and triterpene-saponins. And interestingly, triterpene-saponins are generally considered the main group of phytochemicals which are responsible for both pharmacological activities and side effects of licorice. In this image, you see the metabolic relationships with different terpenoids of licorice. The main compound available in the plant is glycyrrhizine or glycyrrhizic acid salt, which is metabolized into glycyrrhetinic acid or glycyrrhetinic acid monoglucuronide during intestinal metabolism or liver metabolism, and these compounds can act as both active compounds and those compounds which are causing the side effects of the plant, which would be further discussed. So we understood based on the results of virtual screening that licorice is able to somehow inhibit some protein interactions of SARS-CoV-2 virus with our cells and can decrease its infectivity. But aside from all those simulations and computational studies, what real world evidence says? Is there any experimental or clinical evidence suggesting that licorice really acts as something to prevent COVID-19? And that is what we're gonna now take a look at. So I start with in vitro studies. In an in vitro study in very sick cells, kind of kidney cells, the cells were infected with a clinical isolate of SARS-CoV-2 isolated from patients, and the cytopathic effect and platforming efficiency was assessed in simultaneous addition of licorice compounds. This treatment was actually done in three different protocols. In the first experiment, the licorice compounds were added to cell culture, and then after a while, the cells were exposed to the virus. In the second experiment, licorice compounds were added simultaneous with the virus infection, and in the third experiment, the virus was first introduced to the cell culture, and after a while, the licorice compounds were added to the cell culture. Uh, According to inhibitory concentrations, 50% inhibitory concentrations or IC50 values, licorice extract and a GL or the so-called glycyrrhizine were able to inhibit both cytopathic effect and platform efficiency, which are markers of virus infectivity on the cell cultures. And you see that there's a concentration range for both compounds. This is because the three different methods of administration, and it was shown that when the compounds, the herbal extract or glycyrrhizine is added as a pre-treatment, meaning before the introduction of viruses into the cell culture, it would be more effective and the IC50 value would be lower, so it would be more potent in comparison to when it is simultaneously added together with the viruses, and the lowest effectivity was when it was the compounds were added after viral infection. So the sooner the cells were exposed to licorice compounds, the higher efficacy it could show against the viral infection. Another in vitro study, again in very sick cells, showed that lentivirus expressing spike protein S pseudotypes, meaning that just non-infective virus which are just expressing the virus protein. It is not SARS-CoV-2 virus. It is a virus based just expressing a spike protein of SARS-CoV-2. These pseudoviruses were introduced to very sick cells, and upon pre-treatment with glycyrrhizine with 5 millimolar concentration, the infectivity could be inhibited up to 92%, and the inhibition of S protein attachment to the cells were inhibited up to 95%. And another experiment assessed whether the addition of licorice compounds or glycyrrhizine to the virus inoculum would be effective, or whether it should be introduced to the cell culture first. So it was shown that when the virus were first exposed to licorice compounds, they were less actually infective in comparison to when the virus were totally healthy, but the cells were exposed to licorice compounds. So licorice or actually glycyrrhizine were able to both improve the ability of the cells to inhibit viral infection, and was also able to damage the viral structures to some extent, so that the infectivity was reduced. This is another in vitro study, and they assessed a third actually species of Glycyrrhiza inflata. And they used a kind of novel methods for assessing the antiviral activity. In this method, some magnetic beads, small beads, were used, and the spike S1 subunit protein of SARS-CoV-2 virus were attached on these magnetic beads. When these magnetic beads were actually poured into the cell culture wells, a mixture of licorice extract, which was hypothesized to have some compounds which may be linked or maybe a ligand for S1 protein, were attached to these compounds. Then in the second part, a magnet was placed in the well. So because the beads containing spike protein were magnetic beads, they would attach to the magnet. And because some of the licorice compounds, which are in the image shown as the red squares, these compounds were ligands which attach to spike 1 proteins. They also attach to the magnet into the well. In the next step, the wells were washed so that those compounds of licorice um, which were not ligands of S1 protein were washed, and in the last part, the S1 protein subunits were denatured using some enzymes, so that the only compounds, or the only molecules we had in the final solution were those ligands which were attached to a spike 1 S1 protein. And this final solution were exposed to LC-mass analysis so that the structures were defined, which these compounds were. And as you see, 4 candidates were introduced in this part. All these 4 candidates were flavonoid structures. So in this specific species of Glycyrrhiza, it seems that flavonoid compounds were really prone to show antiviral activity via inhibition and attachment to spike S1 subunit protein. In case of animal studies in the next level, there was a study assessing the infectivity of a SARS-CoV-2 protein in a SARS-CoV-2 actually virus in a hamster model. In this study, the hamsters were exposed to virus for 5 days. After this period, they were treated with an oral dose of Glycyrrhiza glabra 130 milligram per kilogram. And after 4 days, the animals were euthanized, and lung viral load, lung histology, lung injury markers, and cytokines profile were assessed. And it was shown that administration of Glycyrrhiza, administration of actually licorice could decrease weight loss, decrease pneumonitis and inflammation, and also the viral load. And the diagram in the B diagram in the image, you see that the red line shows the weight loss change in animals which were in the negative control group. They were just infected with it without any treatment. And the purple line shows those animals, the weight of those animals which were treated with licorice extract, which, as you see, is significantly improved in comparison to the negative control. And the green line shows remdesivir as the positive control group. Also, the histological analysis of animal lungs show that the histological damage to the animals receiving licorice extract is significantly lower than that in the negative control group. Regarding the viral load, the D diagram you see on the right side of the slide show that, again, licorice was effective to improve the symptoms. However, it was not statistically significant because the p-value was 0.08. So numerically, but not statistically, could improve the viral load in comparison to the negative control group. And in another study in rats, it was shown that a diet enriched with licorice extract could significantly decrease the expression of ACE2 receptors in the animals. And as Dr. Zandi said in his lecture, ACE2 is a co-receptor for SARS-CoV-2. So the authors hypothesized that decrease in the expression of ACE2 receptors may be a way for reducing the susceptibility of the animals to SARS-CoV-2 infection. However, this was just at the level of hypothesis. I mean, they did not expose the animals to SARS-CoV-2 virus to see whether really the infectivity was reduced or not. Regarding clinical studies, let's start with this retrospective cohort study from a Chinese medicine. This study was obviously done in China. He Jin Shen Qi decoction is a TCM, traditional Chinese medicine formula, containing several ingredients. Of course, it's a polyherbal formula, not just containing one plant, but one of the ingredients is licorice. So there was a retrospective study in 81 patients with severe COVID-19 disease, which were hospitalized. And in these cases, in comparison to those who only received standard care, the patients under treatment with this TCM decoction had a better improvement in the symptoms of COVID-19, like they had better relief in their fever. And the duration of the negative conversion time of nucleic acid, meaning the PCR test, that famous PCR that I think many of us have done to see if we are infected with COVID-19 or not. So this negative result for PCR was actually achieved in a shorter period of time for those patients received this TCM preparation compared to those who only treated with standard care. Another retrospective cohort study from China assessed another formulation, another decoction of traditional Chinese medicine, which was a mixture of 4 ingredients, and one of them was licorice. So in this case, licorice was one of the major ingredients of the formula. And in 48 hospitalized patients with COVID-19, in comparison to standard care, again, those who took this traditional Chinese medicine decoction could shortly achieve a negative PCR test, and a better improvement was observed in the clinical symptoms of the patients. So about randomized controlled trials, the first study was done in Iran, which assessed a Persian medicine formula. The study included 213 hospitalized COVID-19 patients, and a syrup containing licorice, judo fruit, damask rose, rhubarb saffron was administered as a 10 milliliter syrup every 8 hours for 7 days to patients, hospitalized patients. And in comparison to standard care, this formula could significantly decrease the mean duration of admission for 2 days. It means the patients were hospitalized for less, for actually a shorter period of time, 2 days less than those in the control group. So the formulation in the active arm of the study caused a significant decrease in CRP level of the patients, and it improved oxygen saturation in patients. So it generally improved the symptoms in comparison to the group which only treated with standard care. Another RCT from Iran, which assessed the Persian medicine formula, was a multi-center clinical trial done in 6 hospitals and included 358 hospitalized patients with COVID-19. The protocol of the study, in fact, included 3 herbal formulations, 1 polyherbal decoction and 2 herbal capsules, and the licorice was a major ingredient of one of the herbal capsules. So administration for 7 days caused a significant decrease in the duration of hospital dyspnea for 3 days shorter than the patients in the standard care control group. And it could significantly decrease clinical symptoms of the patient, including dry cough, muscle pain, headache, fatigue, anorexia, and other symptoms related to COVID-19. Another clinical study from Iran assessed the effect of 2 herbal capsules based on Persian medicine formulas on 82 hospitalized patients with COVID-19. And in comparison to standard care, again, the duration of hospitalization was significantly decreased, and the dyspnea and the need for O2 therapy was lower in the patients received herbal intervention compared to the control group. This is also another RCT from Iran, but the formulation NOSCAF syrup was not designed specifically for COVID-19. This is a antitussive syrup, which was also in the market before the COVID-19 pandemic, but during this pandemic, it was also assessed in the form of a clinical study in 124 patients with COVID-19. This syrup contains noscapine and licorice, so it contains two agents, and 20 milliliter of the syrup was administered every 6 hours for 5 days, and it was compared with diphenhydramine as a conventional antitussive product, which could significantly decrease dyspnea and improve cough-related quality of life in patients. So all the evidence we reviewed till now showed that licorice individually or as a combination in a polyherbal formula may be effective to improve the symptoms of patients in COVID-19. So it is not just the claims of a traditional medical doctrine or what we only saw in computational analysis. There are real-world evidence suggesting licorice as an effective treatment for COVID-19. But we should always keep in mind the precautions related to administration of herbal medicines. As you probably know, glycyrrhizin and glyceritic acid in licorice, which are the major compounds, can suppress renin-angiotensin-aldosterone axis and cause pseudohypoaldosteronism, which results in a significant hypokalemia. A case report in Spain reported a female, 82 years old female who was admitted to hospital with a very low potassium level, high troponin T level, and high blood pressure. And the patient was diagnosed with secondary acute myocardial infarction. The image you see shows the infarct area of the patient. And in reviewing the patient drug history, the physicians understood that the patient started taking a large amount of licorice decoction, 1 liter per day, as kind of prophylaxis for COVID-19, because she heard that it would be effective to prevent the infection, and it has antiviral properties and something. It would be definitely important to see to know how much licorice was used for preparation of this 1 liter decoction. But in any how the 1 liter of licorice decoction would be really high dose, and the higher duration of administration would also exacerbate the clinical symptoms and somehow the toxicity of this herbal medicine. So aside from all these beneficial effects of licorice that we saw in clinical and preclinical evidence, we should always keep in mind the safety considerations. And because some of the participants in this webinar might be physicians who may be willing to know about the standard dose of licorice, I put this screenshot of the Botanical Safety Handbook published in 2013. This is the monograph for Glycyrrhiza species, generally Glycyrrhiza species. So it doesn't matter whether Glycyrrhiza glabra or duralensis or any other species. This monograph shows the standard dose, what the standard dose is, and what the safe dose is, and what adverse effects may appear in long-term use and in higher doses of licorice if taken without supervision of a physician. So to conclude, we can say that different levels of evidence, including in vitro, in vivo, and human studies, suggest licorice to be, to some extent, effective in COVID-19. In most cases in clinical studies, licorice was used in the form of multi-component herbal formulas. So one of the limitations of the current studies is that we cannot actually relate the observed clinical response only to licorice, because in case of multi-component products, all those ingredients are doing something, and we cannot exactly explain which ratio of the clinical effectiveness is related to which of the ingredients. So this is one of the limitations we currently have regarding the clinical studies about licorice in COVID-19. Anyhow, because of the clinical, in silico, and preclinical evidence, it seems that licorice can be a future candidate for similar conditions. I mean, maybe other respiratory viral infections. I hope we are not going to face any other pandemic like COVID-19 in future, but there might be other kinds of respiratory viral infections with similar manifestations. And in these cases, based on the available evidence, licorice seems to have a high priority to be considered as a therapeutic option. And again, I would like to emphasize and to keep caution in long-term use and high doses of licorice, especially in susceptible cases like elderly patients with previous history of cardiovascular diseases, and those patients taking cardiovascular drugs because of the possibility of side effects and herb drug interaction in long-term use. So we still need future studies to keep a clear evidence for the safety and efficacy of licorice as an individual or as an add-on therapy to conventional medicinal treatments recommended for respiratory viral infections such as COVID-19. Thank you for your attention. If you want to keep in touch, this is my email address. I will be happy to receive your messages, and I'm here for any questions. So any questions? So I see a lot of our dear colleagues from India and also Iran, and I see the names of our students. I would be happy to receive your feedback. At this part, I would like to ask you to compare the results of these experimental and real-world evidence compared to those in silico studies. I actually, as a researcher in the field of phytopharmacology and phytotherapy, I always face this comment from reviewers when we are submitting our manuscripts. That's how you can extrapolate the results of a in silico study and virtual screening study to humans, how you can be sure, how you can guarantee that you are receiving the same result when you're administering the plant to a human or to an animal just based on the in silico studies. So such gathering of evidence shows that it is not just in silico studies, it is not just computational studies and virtual screening. There are real world evidence that supports some theories based on traditional medical doctrines all over the world. As you see, this plant is highly valuable in traditional Chinese medicine, Indian medicine, Persian medicine, and other parts of the world. So taken together, I think comparison of these evidence along with the in silico studies and visual screening would help us develop further and high quality studies for future cases to treat and to help patients with the final goal of helping the patients. Thank you very much for your positive feedbacks. So no question, no raised hand. No one's gonna give us a comment. So if there's no question, in the end, I would also again ask Dr. Madda if she's going to add any points, maybe. And unfortunately, Dr. Zandi had to leave the meeting because he had to reach to another meeting. I would also be thankful to Dr. Zandi for his participation. Okay, dear Dr. Maddox, would you please?
I want just to say thank you for your administrating these webinars, and thank you for your nice presentations. I have no points. Thank you again.
Thank you very much. The pleasure is mine for having you in this webinar and this joint project, which resulted in publication of a actually high quality paper. In the end, I would like to say thanks to all dear participants for joining us at this webinar, and I would like to invite you to keep in touch with us with the website of Race to Race group in the link you saw in the actually poster of the webinar. We are going to have future webinars in this regard. Okay, Dr. Ismailzadeh, you raise your hand. You can turn on your microphone if you want to add a comment.
Hello, everyone. Hello, Dr. Bahar Solsani. I want to thank you for the very informative webinar. I have no question to add, but I wanted to say that this kind of webinar really covered different kind of trials and different kind of studies which have been done during COVID, which we really actually get through the basics to the in silico studies and also the RCD and experimental. For this almost 2 hours, for the past 2 hours, I think we covered all the necessary points about COVID-19. I just wanted to thank you and all the 2 other presenters for such an informative webinar.
Thank you, Nisha. Thank you very much for your positive comments. Yeah, I hope we can manage to have similar webinars, these forms of somehow bench to bedside webinars, which can discuss different aspects of a disease and how natural products and traditional medicine can help to improve the situation of patients in these cases. And yeah, the Race to Raise link is available in the chat box for your feedback. And if you missed some part of the webinar due to internet the connection, you can view the recorded file. The webinar is recorded, and you can check the recorded file. You can share it with your friends. So for now, again, I would like to say thanks to all your participants and also our dear colleagues at Race to Raise team at the International Affairs Deputy of Tehran University of Medical Sciences. And I hope in any part of the world you are, you can join us for our next webinar. So keep in touch and have a nice day, afternoon, night, and goodbye.